with you my board first, and then after we're going to start our class, right?
Okay.
All right. Okay, my board is visible.
Um,
Uh, Okay,
one
all right.
second. Mm.
Yeah, board is visible or not?
Yeah.
Okay, great. So, the first topic that we have to study on today is the structure of DNA, right?
Yeah.
Structure of DNA. Okay, so I hope because it's your specification of hybrid. So right now, I wanna recall the concept that you have in your previous class about because you have studied this in before as well. So structure of DNA, what do you know about this? Like what is DNA itself? Then after we'll go for the structure of DNA. What is DNA? What's its significance in biology? Aliza?
It's to do with like genes and stuff.
Yeah. Just talk about it. Like suppose as a any if you want to go to tell any layman like this is DNA. What is DNA and what's its significance? Like and DNA is itself what? What's its abbreviation? Like this is what we are going to start from the very scratch. Yeah.
Um,
Deoxyribonucleic acid, right?
oh yeah, and a
Okay.
base and a phosphate.
Yeah.
Alicia, take it
It's
seriously. Put your leg down.
it's it's a deoxyribonucleic acid. So,
Yeah.
can we just talk about its significance first, so then after we can go for to talk about its structure? What is its significance in the body of an organism? Suppose if this is in our cells, what do you think? Why it is important? Suppose there is
Um,
no DNA in our body, so is it possible for us to survive?
because it has the genetic information
Yes, it's a
for
blueprint.
every living
Exactly.
cell.
It's a blueprint, right?
Yeah.
Whatever the information that we have, whatever the activities we are going to do, are whatever the structures which are going to form, that is
Whoops,
triggered
sorry.
by, guided by DNA. All right.
Yeah.
Are you getting this? So this is DNA, deoxyribonucleic acid. Got it?
Yeah. Yeah.
So it's a blueprint. Then after we're going to talk about the structure of DNA. So the structure of DNA, suppose if I'm going to put diagram in front of you, and then we are going to discuss about it, the structure of DNA, right?
Yeah.
So at least I'm going to have a structure of DNA, the very simple structure that we're going to discuss, that is here. Can you see the structure? Is this visible to you?
Yeah.
Okay, so this is the structure of DNA that we're going to discuss. And you have to discuss and to elaborate this structure, what it is, and what are the different components in structure. Definitely, we'll talk about its composition. What are the different components by which this structure is formed, right? So the structure of DNA, here you can see like two strands are here, like this, like
Yeah.
this. Can you see these structures?
Yeah.
Okay. So this is first let me talk about important things like we should as a whole we should understand what is the repeating unit here, what is the monomer here. The monomer that is present in the structure of DNA, that's what we called nucleotide.
Yeah.
Okay, nucleotide. So nucleotide is made up of 3 components. Can you just help me to repeat those components which are the part of nucleotide?
A deoxyribose sugar.
Great. Sugar, deoxyribose sugar. We'll talk about this, like what's its difference. Yeah. Deoxyribose sugar. What's the second component?
A phosphate and a base.
Great. Phosphate is the second component of the nucleotide. And third component that you said, nitrogenous base or base, right?
Yeah.
So this is the monomer or the repeating unit, which is present in DNA. So by the repetition of this structure, the overall DNA is formed. Okay. So right now I've just taken one part of it. Like this is double stranded structure. Like, can you see here the spiral shape structure that's here?
Yeah. Yeah.
Okay. So this is sugar phosphate backbone. Sugar phosphate backbone.
Yeah.
And why we call it sugar phosphate backbone? So that's easy for you to understand, because these two structures, which are going to connect with each other to form a backbone, to form a continuous structure, that's what we called sugar phosphate backbone, right? So in between this, you can see here, definitely we are going to discuss now these different parts, how they are present here in one nucleotide. If I can show you a one nucleotide, I hope you know about this already, like what is the overall structure of nucleotide, then we'll be able to go to discuss about some of the questions that you have in your past paper exams.
Yeah.
So, Lisa, here's a structure that we're going to discuss about. Can you see the structure, Lisa? What is this? What is this structure? Can you see its composition? Like phosphate group is here, pentose sugar is there, then you have a nitrogenous base. What is this structure? Is this not nucleotide?
No.
Then what is
Or it
this?
is.
Then what is this? You have
Oh
just.
wait, no, it is.
Yes,
Oh
it
no,
is.
it is. It is. It is.
Yes, exactly. Because you have explained here, the nucleotide is made up of 3 components. One is sugar, second is phosphate, and third is base. So this is the structure of nucleotide. Okay, this is nucleotide. And this structure is going to repeat again and again. You can see here that we said this is sugar phosphate backbone. Here is phosphate, here is sugar. Again, there will be a sugar and phosphate, sugar and phosphate, sugar and phosphate. So this is how this overall structure is formed, right?
Yeah. Yeah.
So the one part that we have discussed, that is sugar phosphate backbone. Then after we can go to talk about its other components. You can see here base pairs, right?
Yeah.
We have nitrogenous bases. So in nitrogenous bases, you have 4 bases, which are the part of this DNA structure. One is, can you please help me to repeat? One is
Guanine.
guanine. Second is
cytosine,
cytosine. Third is
thymine,
thymine. And the last one is
adenine.
adenine. But the important thing that you need to understand here, like how these nitrogenous bases are going to combine with each other. This is what we need to understand. Which nitrogenous base is compatible to the which type of nitrogenous base? This is what we need to talk about. Guanine, cytosine, thymine, and the last one is adenine.
Yeah.
The structures overall, you should have some recognition. Somewhere they ask you this question, like somewhere they'll give you different structures, and in your past papers, so you should be able to understand to recognize these nitrogenous bases as well. We'll talk about this as well, because as I have seen in many of the past papers where they ask question, they have given some structures, and those structures, student is there to student has to identify which structure is present there, like what type of structure it is. So in this case, first we need to understand, like, as we know, these are we used to divide into two main types to all of these four nitrogenous bases. So can you just please help me to recall that the division of these nitrogenous bases?
Guanine goes with cytosine.
I'm talking about the pyrimidine and purines.
Pardon?
Do you have an idea about how to differentiate these two structures?
What do you mean?
Let me show you. Let me show you for you.
Okay.
And you need to remember this as well to idea. You must have an idea, because somewhere you need to identify this. We will discuss some questions so you will understand this. Here it is. Look at this. Can you see this structure?
Yeah.
And this is the classification of all of these nitrogenous bases. One is uracil that we will discuss in RNA. Right now we are talking about the DNA structure, so that's why I didn't talk about that. So purine and pyrimidine, these are two main structures. All right.
I don't think we've done that. I don't think we look on that.
You should know about these types here. This is how we can divide them: adenine, guanine, cytosine, thymine, uracil. So these are always classified under these two classification, these two types. No need to go for further detail, right? Just to know about purine and pyrimidine. And in different types, like you should know adenine and guanine is present in purines, because these are double ring structures, and pyrimidines, these are the single ring structures. This is what identification you should know at your level, right? So then after we'll talk about its bonding, Lisa.
Yeah.
And guanine is going to make a bond with cytosine, and thymine is going to make a bond with adenine. And do you know the types of number of bonds which are present there? Yeah.
2.
In which?
Yeah.
Which
wait,
is there to? Yeah, just recall it if you have an idea. And what type of bonding are present there? What type of bond is there?
Hydrogen bonds.
Yes, hydrogen bonds are there. So you can see in many of the cases that we will understand, yes, these are the hydrogen bonds. And suppose I can have a structure for you. Like here we have a structure that is showing you the bonding between these two. In adenine and thymine, you will have double bond or triple bond. That's a double bond, right? So
Yeah.
in a similar way, you will have a triple bond in case of guanine and cytosine. So here's a structure that's showing you like this is a double bond present here. Can you see this?
Yeah.
So this is a double bond and it is like here. And on the others two parts that we have the two nitrogenous bases, that's guanine and cytosine, they contains always triple bond. So adenine and thymine, they contains double hydrogen bonds or two hydrogen bonds. Okay.
Yeah.
I'm just showing like this. And guanine and cytosine, they're going to have triple or three hydrogen bonds.
Okay.
Okay. So this is the overall structure that we should know. As far as further, like you can see, here we have, these are the bases. We have discussed about the sugar phosphate backbone. So another important thing, like we should understand that this structure is anti-parallel.
Oh yeah. Mhm.
Anti-parallel. I hope you know about this anti-parallel.
Yeah.
Anti-parallel is definitely the two strands which are going to opposite to each other. So
Yeah.
opposite structures, just explain it for me. Yeah. What is in your mind how this structure is like, how
It's
it is anti-parallel. So by
because these two strands run in opposite directions from the deoxyribose sugar at the 3' end, and then at one end to the phosphate 5' end at the other end.
yes. So this is how you can see in this structure that's in front of you. It's 5 to 3 and 3 to 5. The opposite structures. Like you can see here in this diagram that's showing you how these DNA strands are anti-parallel to each other. So can you see here in this diagram, it is started the one first, this is from one side. You can see here the one nucleotide is going to form a bond with the other nucleotide. That's to the phosphodiester linkage. This is phosphodiester bond. This is what also you also keep in mind. Phosphodiester bond. And this bond is formed between the nucleotides. So, and the second thing is, like you can see here, this is from 5 to 3 direction. Like here you have oxygen on this side, and then you are going on this, in this direction, this deoxyribose here. On the other side, you can see here, it's from 3 to 5 direction, right? So it's a double stranded DNA, which is opposite to each other. So here you can see the hydrogen bonds, which are double bonds between guanine and cytosine, and thymine and guanine. You can see here, like that's a bond between which is present between a pyrimidine and purine. So are you getting this?
Yeah.
Okay. So now I'm going to have one more important thing for you. Just a moment, please. Stay with me.
Okay.
I'm going to have a patient in front of you, so we'll discuss it together. Eliza, look at
Yeah.
this. A diagram shows the structure of part of the nucleotide. You can see here a subunit X and prime and Y. Which row in the table identifies subunit X and prime and Y? So you have options here. A in nucleotide, prime and Y 3 and, okay. And B is amino acid 5 prime C, amino acid 3 prime, D, nucleotide 5 prime. So what do you think? According to this discussion, and the diagram shows the structure of part of DNA molecule. So what is subunit X here?
It's a nucleotide.
Okay, it's nucleotide. So
Yeah.
this prime and Y,
It's the 5 prime.
great. So which option is correct here?
D.
Great. So option D is correct, okay? In a similar way, we have another question. Like I'm going to have 2 questions for you more for this.
Okay.
Mm, This is what you will study here, but it's going to a list describes feature of different chromosomes. We are not talking about chromosomes right now. So we can have metabolic pathway. Mm, Just a moment, please.
Okay.
Yeah, here's a structure that I'm going to talk about. Suppose we have a question here. That's I'm going to paste for you. Eliza,
Yeah.
look at this, please.
Okay.
At the start of it's a replication. The double helix unwind and separate as shown in diagram. No, leave this. This is not for your specification. Because right now we are talking about this is from DNA, but we are not talking about the overall its structure. So that's why there's no need to go for this. So I have some other questions for you, so I'm going for that.
Okay.
How much? I mean, when you started your this preparation, like when you're going to appear in your exam.
Yeah.
When are you going to appear in your exam? This is what I'm, Lisa.
When am I going to sit the exam?
No, I'm just asking about like you have started now. I mean, yes, these codes are in
Yeah, we've
which
started the course.
you started the course. So
Yeah.
so when you're going to appear?
What do you mean?
I mean,
Appear for what?
exam, your final exam.
I'm not sure. We've not got the dates yet.
Approximately time. That's what I just.
Oh, it'll be May next year in May.
Means May, June 2026.
Yeah.
Okay. All right. So I have some other questions like to. You should know about this. Just me check some questions. Yeah. No, I'm not going for. Just a minute. Sorry, it's taking time. So here's a question that we need to discuss about. So, Elisa, I'm going to have a question like this one. Is this visible? Elisa, can you see these questions?
Yeah.
And you have the first question: the DNA molecule contain polynucleotide strands. Individual nucleotides are joined together to make a polypeptide strand. What type of chemical reaction takes place when two nucleotides in a single polynucleotide strand are joined together? Yeah, you can think about it. Like to discuss, I'm going to discuss with you as well. Just any guess about it. Then after we're going to talk about it as a whole. Yeah. Any guess from your side? Hmm? Mm-hmm. Is this like you should think about condensation and hydrolysis. So if I'm giving you the options, like I'm going to give you two options, whether it's condensation or hydrolysis. That's a new concept that I hope you know once this is what you have studied
I
somewhere.
don't think is this I don't think we don't talk about that.
You
We
didn't
don't we is this in like Scotland? Is this higher questions in Scotland? Because we don't like talk about those.
you
We
didn't
never
talk about this. No, these
no.
these are the part of your past paper exams, right? So but you should know as a whole like what type of like suppose name the chemical released when the bond is formed between the two nucleotides. So we just need to talk about it. DNA molecule contain two polynucleotide chains. Describe how these two chains are held together. Okay, let's talk about this last question. We'll get back to that as well. The last question. Let's talk about this. Like if you can solve it from your side.
I can't see it. I think you need to scroll down a bit.
No, that's on the board. Here it is. Can you please just can you follow
Oh wait,
my
I
cursor?
forgot.
Can you follow my cursor? Here it is. Visible?
No.
The last question, Aliza.
Yeah, I
It,
can't see it.
you can't say it. It's just scroll down. Just follow my cursor, please. Follow
It
my
doesn't
cursor. There's a
it doesn't
question. A
let me.
DNA molecule contains two polynucleotide chains. Describe how these two chains are held together. This is a question. It's a 3-mark question that can you say it now?
No.
How is it possible, Eliza? Okay, just a moment. Where, where is your cursor? You can like this one. Is this not visible?
No. I think you need to just put it up a bit more.
Hmm. I mean, I'm here right now. I mean,
Does it not let you put it up?
I'm just
Oh yeah,
pasting
I can see it. I can see it now.
this one.
Yeah.
So here's a question. A DNA molecule contains, it's what I'm going to help you here. We will discuss about this as well, but keep in mind, the chemical reaction that's a hydrolysis. That's the presence of, sorry, in this case, yeah, it is because we are talking about the reaction which is going to carried out when these nucleotides are going to join to each other. So that's condensation.
Okay.
We'll discuss this as well. And the molecule which is going to release when this bond is formed, that's water. Okay, so because this condensation process is actually the release of water, leave that. Talk about this one. The DNA molecule contains two polynucleotide chains. Describe how these two chains are held together. So what do you think how these, we have talked about this, how these structures are going to held together? I mean, they are together here. You can talk about the first thing that you can talk about that is phosphodiester bond in backbone. Right down here in the answer. Eliza, can you
Yeah.
please just write down here? take a pen from here. So this is because you have to contribute here. I mean, at this point of time, you have to understand the pattern and the style of exam, the pattern and the style of question that you will get in your exam, right? So
Yeah.
our main focus would be like here to just practice on these questions. Whatever the topic you will study, you have to practice on those questions so that you will understand what type of questions that you will get in your exam, right? So the first thing that you need to write here is phosphodiester bond. So from where you can start, can you please just write here in the lines
Yeah.
given here? Yeah.
Yeah. It might be a bit, I can't really write it properly, sorry.
It's okay. At the start, you will be, but after that, with the passage of time, you will just, most of the students, they get, I mean, they habitual and somehow they used to for this
What
writing.
was it? Phosphate.
Phosphodiester bond, which is
Oh, we've
present
not looked.
in the bond,
We've
in backbone.
we've not looked at that. I've never heard of that.
Phosphodiester bond in the backbone.
Yeah, we've not looked at that. Oh, whoops, that was really bad. Sorry. I don't know how to spell it.
Let me show you.
I don't know. It's because we've never looked at it.
PHOS, PHO, PHOS, PHO, yeah, DIESTER. No worries. DIESTER.
STER. Sorry, it's not neat at all.
It's okay.
Can't do it properly.
Bonds in backbone. No worries, no worries. This is very common. Bonds in backbone. Right? This is one thing you need to talk about. But this is what you have to explain it properly. Like the phosphodiester bond is present between the nucleotides in the, we have discussed about this, the phosphate and sugar backbone that we have described here in the diagram there. Okay? This is one thing that you need. And the second point that you need to add here is hydrogen bonds, hydrogen bonding. Because hydrogen bonding is present between the nitrogenous bases. So this sort of bonding has also, hold the structure of DNA together, right? This is the second point that you need to add. Okay?
Yeah.
Okay. And then the third thing that you need to discuss here is adenine bonds with thymine and cytosine bonds with guanine. This is the third point. Okay, but this is just a hint that I'm just giving you the points like you should write in this domain. You have to write the proper sentence about it.
Okay.
Just write on the third one as well. Cytosine bond with thymine, no issue. I mean, write down as much as you can. It's okay. With the passage of time, you will understand and I'll improve you in all of that. So my concern is like you should be able to understand the style of questions that you will get in your exam that's specific to you. Like we have to discuss about its specification. You already know what's your national biology course that needs like it. You should understand what type of questions are there, right? Have you just read out your course specification? What is the requirement? What type of questions that you will get there?
Yeah.
I mean, how many types of questions that you'll have in your course?
In total, wait, let me check. Hold on, it's just loading. I think it's 25 multiple choice, and then
that's I know, but I'm talking about the types of questions, the style. Let's suppose, I mean, knowledge-based questions, understanding-based questions, like your mathematical skill-based questions. This is what I'm asking. You should know about these types of analytical questions. Yeah.
Yeah, I think it's like 75 it's like 75% knowledge questions, and then it's 25% problem solving questions.
Problem solving questions. So you
Yeah.
should prepare yourself according to this course specification, right? So so we'll discuss about the problem solving questions as well. Somewhere you will get, and that one problem solving question would be from this part, from this DNA structure. They'll give you some percentage of the nitrogenous bases. Like they may give you the percentage of guanine and cytosine. And they may ask you the question like, what is the percentage of for example, I'm saying the adenine and thymine, if they're giving you the percentage, then he'll ask you the question, just tell me the percentage of cytosine there. So that's a tricky question that you need to understand. We'll talk about this as for instance, like we'll go for that as well. But here's another question that I'm going to talk about. So Aliza, look at this.
Yeah.
Is it visible? Just below this.
Yeah. Yeah.
I've just pasted here. Okay. The question here is, DNA is a biological molecule that varies between individuals. Sections of DNA code for proteins. Shows the structure of part of a DNA molecule. The circled region shows one nucleotide. Name the components of this nucleotide. That's very easy. You have already explained. Just write down.
The circle. Or should I write it down, or should I just say it?
No, you need to write because I have to help you to write, because you know most of the students, they are very good in verbal discussion, like they used to give answers verbally, but unfortunately whenever I take any assessment and to say, they are very poor in writing. So your concern should be writing practice, how to write the answer properly in that specific space. Okay, this is what we need to learn as well. All right.
Okay.
Okay, yeah, you just need to write here.
Okay. Oh, that's the whoops. Circle.
Yeah, circle. Like circle is. Good. Phosphate. Great.
Sorry, it's really hard for me to write.
No issue. This is your first time, so that's why you're feeling like you're facing like it's difficult to write. But I have observed you can also use any tab here as well. Like just like myself here, I'm using this. Can you see it? Like here's a tab and the pen that you can use here. If you can find some writing pads, so it's also easy for you to write. Suppose I can write easily as compared to you, but with the passage of time, you can also learn this how to write by using this. Okay.
Okay.
Like I'm just writing the normal, like as phosphate, a normal person as you write. Okay. So yeah, this is what is the last point? Pentose sugar, deoxyribose. Okay.
Yeah.
Good. So one more question that we're going to talk about. I'm not giving you just for your judgment. This is for definitely I help you because we have just studied this all, so that would be easy for you to go through this. So show this figure is showing you. Can you see this here?
You just need to put it a bit up.
This one. Aliza, this one. Is this visible?
Yeah.
Just above this. Okay. Can you see it? What is this?
It's nucleotides.
Yes, and words. The question is, That's a 2-mark question that you need to attempt here. This one. Can you see it? Just below
Draw
this.
and label the bonds.
Draw and label the bonds holding the nucleotides together as part of the DNA molecule.
Oh, okay. So it would be.
Hmm.
Oh, sorry, I just moved it.
No worries.
Like that.
Hydrogen bonds.
Is it
But this is
another?
not hydrogen bond. If you'll write this, if you'll draw this hydrogen
it?
bond, so your exam will not give you marks.
Is that it? I don't know.
No, in dots. In dots.
Oh, and dots.
Yeah.
Wait, so like. Oh, it's squinty. Sorry. Like that? No.
Which one? Yeah, it's okay. Yeah, that's fine. That's fine. But I mean, just dotted line. Nothing is difficult. Like hydrogen bond is just we always represent this by like this. Okay. So this is. So you have cytosine and guanine here. How many hydrogen bonds are here? Draw and label the bonds holding the nucleotides together
Is it
as
two?
part of a DNA molecule. Yeah. Yeah.
Is it two bonds?
In case of cytosine and guanine, not adenine and thymine.
Oh, cytosine and guanine is a triple bond, and then
Yeah.
adenine and thymine is a double bond.
Yes. So you need to and just name them as well because you have to label them. Write the name is hydrogen bonding. Okay, Lisa, are you getting?
Yeah.
But just draw them properly. Like just consider this like you are doing this in your exam.
So
Guanine and cytosine, you should have 3. Like you should show 3 dotted lines, Lisa.
Oh, right. Okay.
Got it?
Yeah.
3 dotted lines. And in a similar way, you should add 2 dotted lines in case of adenine and thymine. Good, but this is your first time. It's okay.
Yeah,
All
I
right.
can't do it properly.
Oh, okay,
So.
okay. But you should remember, like these dotted lines must be there in 3 dotted lines in case of cytosine and guanine, and 2 in case of thymine and adenine. And can you just please write the name of it? Should I write? Because
Yeah.
hydrogen bonds, okay, are hydrogen bonding. And I mean, you have to show the bond here between the nucleotides as well. Like this is what you have shown here between the nitrogenous bases, but there is a bond which is present between the 2 nucleotides as well. That's a phosphodiester linkage.
Yeah.
Yeah. How you can show that? Yeah. Yeah. Good. You should try. I mean, you need to show this bond which is present in the backbone of it. Phosphodiester linkage. You have to connect like 2 nucleotides together. How you can do this?
Like
Phosphodiester.
I don't know. Is it? I'm not sure.
Okay, you know what is this? This structure is what? This is phosphate. This is phosphate structure.
Yeah.
And it should there to no, not this. Aliza, it should be like this here. Okay, because this is carbon.
Oh yeah.
Right? So this is how you
Yeah.
have to show it. And in this case, again, you need to show how it's going to make a structure. Like this should be here. Okay. So this is, can I label it?
Yeah.
Phosphodiester bond. All right. So this is what you need to understand. Got it?
Yeah.
All right. So this, there are many other questions. I used to give a worksheet at the end of the topic. So right now we have covered a few questions from this specific topic. All right. So, should we conclude this for today, Aliza?
Yeah,
Everything is.
yeah.
Are you
Yeah,
understood?
thank you. Yeah.
Okay, that's great. So, Lisa, I'm going to conclude this now. You can over to Sir Asim. He wants to discuss.
Assalamu alaikum, Aliza. How are you?
Yeah, I'm good.
Thank you so much, Saramjit. Okay, Liza, let me move you in
Thank
my
you.
room.
Thank you, sir.
Thank you.